everybody. Can you hear me? I'm Betsy Boughton, and thanks so much for joining us today, either on Zoom or in person. Today, I'd like to introduce Kyle Kirejevas. He's a research intern at the ranch in the agroecology program, and he's been here since about May. So 5 months. He's done quite a lot in 5 months. He graduated with his bachelor's from University of Miami in biology with minors in chemistry and ecosystem science and policy just in May. And then he came directly to the ranch. And at Miami, he worked as a lab assistant in the FCOMI lab. And he's really interested in microbiology, and he studied the role of the microbiome and its interaction with the hydrology and how that affected tree growth and health in the Everglades. So when he came to the ranch, he really wanted to work on microbes again, and he's super interested in climate change as well. So he was able to take advantage of a new experiment we have at the ranch that's manipulating precipitation and creating drought and flooding treatments. So now I will, after the internship, Kyle's going on to work at PACE Analytical Lab in Tennessee. And we really are so thankful to Kyle for all of his contributions at the ranch during his internship. So thank you, Kyle, and I'll turn it over to you.
Awesome. Thank you so much. All right. So I'm going to be talking about the effects of precipitation, land management, and grazing intensity on soil microbe communities and soil characteristics today. So the first thing we should talk about is climate change and predicted changes in rainfall. So there should be increasing climate variability. It's one of the most consistent changes forecasted by global climate models. Overall, the study region is projected to evolve towards a drier future in 2100 under high emission scenarios, which is RCP 8.5 from the IPCC Fifth Assessment, which is the scenario that our planet is currently transitioning towards. There's also variability in regards to wet and dry season precipitation. That tends to be highly uncertain. There's slightly greater percentage of models in Central Florida specifically that predict increases in dry season, but decreases in wet season precipitation. There are also forecasts of increasing heavy rainfall events and elongated drought spells in the future. But the precipitation treatments in this experiment included allow us to capture the full range of plausible yet highly uncertain future changes in precipitation variability. The next thing I will talk about is some of the importance of microbes. So microbes such as mycorrhizae improve the ability of plants to uptake water and nutrients. Beneficial nematodes can help combat potential threats to plant health. Bacteria can help with processes such as decomposition, ammonification, nitrogen fixation, and nitrification that help make nutrients bioavailable to plants. So with changes in precipitation, microbes can respond through changes in physiology and community structure. These changes in community structure can include changes in just the total biomass or the composition itself. And these changes can then go on to alter the rates of nutrient cycling and availability, as well as soil fertility. That's because different microbes will have different capacities to perform these processes. Changes in nutrient cycling and availability, as well as soil fertility, can then go on to impact plant growth and performance. And whether these plants serve as crops or forage in agroecosystems, their growth and performance is very important to food supply, especially as there's an increasing need to understand how the food supply will be affected with the threat of food security that comes with climate change. So by examining if cattle grazing or land management can offer resilience to detrimental alterations or foster beneficial alterations in soil microbe communities, we can move forward with strategies that could allow us to better adapt to climate change and the issues that coincide. And because of the aforementioned high variability in terms of future climate of this region, these ecosystems here are potentially some of the most vulnerable, making adaptation strategies more critical to understanding. So now I have some relationships between soil microbes, precipitation, land management, and grazing intensity. I know this slide is very wordy. I'm sorry about that, but all this information is very important for my hypotheses that will soon follow. So for precipitation, soil microbes are very sensitive to changes in moisture, and their activity tends to decrease as soils dry. Increased precipitation has been shown to increase microbial abundance, but has not shown significant changes to the composition of the community. Drought itself has been shown to cause changes in that community composition and structure. Bacteria will become increasingly more dominant over fungi in drier conditions. Gram-positive bacteria can survive better under environmental stressors such as drought due to thicker cell walls and the ability to form spores. And the diversity of microbe communities also suffer from drought. So for land management, the pasture types can impact microbe diversity through the presence of plant diversity, and that plant diversity can then also be impacted by grazing. Previous studies at Buck Island Ranch have shown that long-term land use intensification increase the biomass within microbial communities while reducing the relative abundance of fungi, and improved pastures have been shown to have a higher proportion of gram-negative bacteria compared to semi-native pastures. And for grazing intensity, with increased disturbance in the form of grazing, the bacteria will become a higher proportion of the soil microbe community. A previous study at Buck Island Ranch has shown that with higher intensity grazing, that will result in a higher amount of microbial biomass. And the proportion of gram-positive bacteria has been shown to decrease with increasing grazing intensity. So there are some gaps in knowledge I would like to address. Currently, research on experimental precipitation manipulation is dominated by other biomes such as temperate, arid, and semi-arid regions. So subtropical biomes are very understudied in this regard. And then furthermore, on top of that, research for the effects of experimental precipitation manipulation on soil microbes is even less abundant in that sense. And overall, there's also just a lack of research on the combined effects of grazing, precipitation, and pasture type on soil microbes. So the first thing to understand with this project is the project that it stems from, which is the Rainout project at Buck Island Ranch. So this was a project funded by NIFA to Dr. Zhengxiao Zhu at UF in collaboration with Dr. Betsy Boughton. And there's two main overall objectives of this project. One is to examine interactions of different grazing regimes and land management on the multifunctionality of subtropical grasslands under rainfall variability, and examine the extent to which grazing regimes can serve as an adaptation strategy to enhance climate resilience of grasslands utilized for cattle production. Within this experiment, there are 2 pasture type treatments, 2 grazing treatments, and 5 precipitation treatments. And it's also important to note that this project has only been running a year since 2021. So like I said, there's 2 pasture type treatments. Improved pastures are grasslands that have been converted to non-native C4 forage grasses, namely Bahia grass, and the semi-native pastures contain a mixture of native grasses, such as some Andropogon species and some Panicum species. And they also have the non-native C4 forage grasses, such as Bahia grass. There is a higher cattle load in these improved pastures. As you can see in this chart, improved pastures are nitrogen fertilized and they have a legacy of phosphorus fertilization, while semi-native pastures have never been fertilized. There is liming in improved pastures, but not in semi-native pastures. Seeding and ditching are present in both pasture types, but there is more of both in improved pastures. So now I'll talk a little bit about the project design. So there are 8 sites within the Rainout project. You can see in this map on the right, the green areas represent the semi-native pastures and the orange represent the improved pastures, and where the stars are the different sites for the Rainout project. As you can see, there's 8 divided, 4 within the semi-native, 4 within the improved. And there is a block design and it spans a hydrology gradient with each block containing 2 sites, 1 semi-native and 1 improved. And within each pasture, as you can see from the way I've shown it, that 1 star is going to be represented by the purple box on the left. That would be one of the pastures. And then within that, we have the green box, which represents where the Rainout pasture would be. And within that, we have those 2 red boxes, each representing one of the grazing treatments, whether it's high intensity grazing or low intensity grazing. And then within each of those red boxes, we have the 5 different precipitation treatments. And this randomized block and hierarchical experimental design allows us to test the effects of interactions. So within this, there are the 5 different precipitation treatments. As I mentioned, I won't get into the numbers too specifically. I'll just be very general about it. If you have questions, you can ask later and how we got those numbers. But for the increased quantity precipitation treatment, the wet and dry season both get wetter. In the reduced quantity, the wet and dry season both get drier. In the enhanced seasonality, the wet season gets wetter, the dry season gets drier. The inverse is for the diminished seasonality, the wet season is drier and the dry season is wetter. And then the control is basically as it is and there's no treatment. So now for my hypotheses and objectives. So my first hypotheses relate to microbial biomass. So the precipitation, which is the first one, is going to follow trends that we see that are expected for the soil moisture within the Rainout project. And like I said, studies showing that with increasing moisture, microbial abundance will increase. So the increased quantity is expected to have the highest microbial biomass, followed by enhanced seasonality, then the control, then diminished seasonality, then the reduced quantity. For the improved pastures, we expect that to have a higher amount of microbial biomass compared to semi-native pastures based on previous studies at Buck Island Ranch. And for grazing intensity, we expect high intensity grazing to have higher microbial biomass compared to low grazing based on those previous studies at Buck Island Ranch. For my second set, we have relating to the relative abundance of soil fungi. So part of this will be based on the soil moisture trends that we see. We also have been told with increasing moisture, the community composition didn't change too much, but with drought, it did change. And we know that soil fungi tend to like environments that are more wet. So we're expecting a lower relative abundance of fungi as it gets drier. So we're expecting increased quantity, enhanced seasonality, and control to be relatively the same in terms of the relative abundance, whereas those will all be higher than the diminished seasonality, which will be higher than the reduced quantity. We're also expecting semi-native pastures to have a higher relative abundance of soil fungi compared to the improved pastures, and that comes from the previous study at Buck Island Ranch. And then we also expect low grazing to have a higher amount of relative abundance of soil fungi compared to high grazing. And that comes from studies showing that disturbance such as grazing promotes a higher relative abundance of bacteria. So there would be less fungi and in the high grazing and more fungi within the low grazing. The next set of hypotheses is about the relative abundance of gram-positive bacteria. So the precipitation hypothesis follows the inverse of the expected soil moisture trend because, as I mentioned before, gram-positive bacteria tend to do better with drought. They can better handle that environmental stressor from drought because their ability to form spores and their thicker cell walls. So the reduced quantity is expected to have the highest, followed by diminished seasonality, followed by control, followed by the enhanced seasonality, followed by the increased quantity. We expect the semi-native pastures to have a higher relative abundance of gram-positive bacteria compared to the improved pastures. And then we also expect low grazing to have a higher relative abundance of gram-positive bacteria. And that is based on studies showing that the proportion of gram-positive bacteria tended to increase with less grazing, less intense grazing. And my last set of hypotheses are about the microbe diversity themselves. We expect this to also follow the same trends for soil moisture. And then for the pasture type, we're expecting semi-native to have more microbe diversity than improved pastures. That's because we have unpublished data from Dr. Betsy Boughton showing that semi-native pastures have higher plant diversity, and we've seen that there's been a positive correlation with microbe diversity and plant diversity. And then, as I said before, that high amounts of grazing can remove some of the plant diversity. So with less intense grazing, we're expecting less removal of that diversity within a pasture. So that would have more microbe diversity with the less intense grazing. On top of this, I also had some other objectives. I wanted to explore how the interaction between treatments impact microbial biomass, the percentage of fungi within soil, and the percentage of gram-positive bacteria within soil. And then I also wanted to explore how these treatments and their interactions between them impact soil nutrients, soil pH, and soil organic matter. So now for my methods, here is the study site. You saw this before, this map on the right, and I circled the pastures that I included. I did 6 of the 8 pastures that were involved. Half of them are improved, half of them are still semi-native. And 4 of the chosen pastures were matched for the hydrology gradient that I mentioned before. So there's 2 complete blocks there, but 2 of the chosen pastures were not matched. And that's because a paired pasture for one of them was more recently burned, and a paired pasture for another had a lot of standing water, and we figured that this would introduce more variability into the results. So now for some of my process. So the first thing I did was collect soil by coring it. And then from there, I would sieve some of it. And then with some of the unsieved soil, I would measure the pH of that unsieved soil. And then I also took some of the unsieved soil and sent it to Ward Labs for PLFA analysis, which basically tells us about the microbe community composition. So from the sieved soil, one thing we did was a double acid extraction. And then we used malachite green in order to get diammonium phosphate. And then we also, with some of that sieved soil, did a potassium chloride extraction. And then one of the things we did with that potassium chloride extraction was we used hypochlorite to get ammonium. And then we also used vanadium chloride in order to get nitrate. Another thing we did with that sieve soil is we dried it. And from there, we got the soil moisture. With that dried soil, we then ashed it and then got the soil organic matter. And then from that ash soil, we did an aqua regia extraction. And then we then again used malachite green, but this time to get the total phosphorus. And then we also used some of that sieve soil and sent it to the Waters lab in Georgia for further chemical analyses for things we couldn't do at the lab at Buck Island Ranch, such as looking at aluminum levels in the soil. So here are some pictures that are just going to show that process. The first picture is me coring some of that soil in one of the pastures, then sieving it, then doing some of those extractions, then plating that with some of the reagents that I talked about before. And then those are what those plates would look like that we would then analyze in order to get the concentration of different nutrients. The one on the top right is an ammonium test, and then the one on the bottom left is a nitrate test. So then for statistical analyses, for the data that was normal or could be transformed to be normal, we did a linear mixed effects model. And with the linear mixed effects model, all the treatments are examined. And when there's no significant interaction is found for a treatment, the model is then simplified by removing that treatment. And then eventually it provides the model that best explains the data. And then for the data that was non-normal and couldn't be transformed to be normal, I did a Kruskal-Wallis test. And that's just used when the assumption of normality is violated. It uses a summed rank design similar to a Mann-Whitney U test, where the values for the response variable are ranked numerically from smallest to largest, disregarding which treatment it came from. The ranks are then averaged for each treatment, and it is determined if those average ranks are statistically significantly different. Like I said, it's similar to that Mann-Whitney U test, but the Kruskal-Wallis test can accommodate more than 2 groups, which at some point I had 20 to look at. So yeah, and then I also wanted to mention one of my soil samples out of the 60, which was for a semi-native pasture with high grazing and the increased quantity precipitation treatment. It was not sent off to Ward lab for a PLFA analysis, so it was not included within the data. So now for some of my results. We found that the improved pastures have a higher microbial biomass. We got this through doing the mixed linear effects model. And after running the model, it was shown that the pasture type was the only treatment that had a significant effect. And like I said, the improved pasture had more, and the p-value was 0.00018. The next thing I looked at was the relative abundance of soil fungi, and none of the treatments actually had a significant effect on the relative abundance of soil fungi. The p-value was about 0.1. And as you can see here, and that was for the pasture type, which was the closest to being significant. And then on the right, I have this stacked bar graph. And then with that, you can see bacteria were much more abundant than the fungi. And while it is generally expected that bacteria would be present in a higher percentage, and even more so in the dry conditions where bacteria will dominate, I think it's important to note that fungi are usually desired and often considered indicators of good soil health. So next, we looked at the relative abundance of gram-positive bacteria, and we found that the semi-native pastures had that higher relative abundance of that. As you can see, the p-value here was about 0.003501, and the percentage or the stacked bar graph here shows you that the semi-native did have slightly bit more. These were very gram-positive dominated, and this makes sense because we had a very dry wet season and there's high temperatures. And like I said, the gram-positive bacteria can deal with those environmental stressors better and survive better in those conditions. So next we looked at microbe diversity, and once again, it was the pasture type that showed a significant effect. Improved pastures had a higher microbial diversity. The p-value was about 0.02. And then for some of these different treatments, I have graphs later. If anyone wants to ask about them, I can show them, but I didn't include them in this section. They were mostly non-significant. But next, for some of our objectives, we looked at some of the soil characteristics. So for the phosphorus concentrations, we did another linear mixed effects model. This was able to be normalized by doing a log transformation. And we found that the improved pastures had a higher concentration of phosphorus. And this makes sense because, like I said, in the pasture type differences, that Buck Island Ranch has a legacy of phosphorus fertilization in the improved pastures. We did it until 1986. So this is kind of an expected result. And then on the right, I showed that there was a bit of a correlation with aluminum in the soils, and that makes sense because aluminum is what is binding the phosphorus in those soils. So next we had a pretty interesting result when we did the linear mix effects model for the soil organic matter, and it found that both pasture type and grazing intensity individually had significant differences. So for the pasture type, the improved pastures had higher soil organic matter, and the high grazing intensity also had higher soil organic matter. And the p-values are listed there. So I also looked at soil pH, and we saw that the improved pastures had a less acidic soil compared to the semi-native pastures, and that was statistically significant. And this also makes sense based on the history of the pasture types. The improved pastures are treated with lime to reduce the acidity and to improve soil fertility. So we expected to see this result. And then I also wanted to show this interesting correlation between soil organic matter and microbial biomass. There isn't a super strong correlation, but there is a bit of one, and this makes sense because the soil organic matter usually serves as food for the microbes within the soil. And I just talked about a lot of results, so I made a little table with a little key. So the ones that say no hypothesis, those were just kind of objectives, and we have either if it was significant and supports the hypothesis or doesn't support the hypothesis. So I'll show these for microbe diversity or microbial biomass. As I said, that pasture type had an effect, which kind of is in line with that hypothesis that I had for the pasture type, whereas the other two individual treatments didn't have a significant effect as I predicted, and the interactions didn't have effects. For the relative abundance of soil fungi, none of the individual treatments had an effect as I thought they would, and none of the interactions had effects. For the relative abundance of gram-positive bacteria, the semi-native pasture had more gram-positive bacteria as I expected with my hypothesis. The other individual treatments had nothing. The interactions had nothing. For microbial diversity, this was the interesting one because there was a significant result, but was in the opposite direction of my hypothesis. And then the individual treatments and interactions for the other ones didn't really have any impact, no significant changes there. For the next three, phosphorus, this was one of my objectives, so there was no hypothesis, but the pasture type was the only one to show in a significant effect. For soil organic matter, pasture type and grazing intensity showed significant effects, and for soil pH, the pasture type showed a significant effect. So now I'll do my discussion, future directions, and conclusion. So individually, the precipitation treatments, as well as the grazing treatments, with the exception of soil organic matter, did not show any significant differences within the microbe communities or soil characteristics. I think perhaps if this treatment or if this experiment had been going on longer or has been done in the future after the Rainout project had been going on longer, where the treatments maybe had more time to actually show an effect, we may see different trends. And like I said, the pasture type showed no significant effect on the relative abundance of soil fungi. And I think, as we expected, and with previous studies, Buck Island Ranch showing that. And I think this may have been due to the relatively dry wet season, diminishing the impact of the less extensive ditching in semi-native pastures, preventing them from kind of holding more water, which the fungi tend to like. And the improved pastures showed higher microbial diversity compared to semi-native pastures, which was in the opposite direction that we expected. And I think this may have been for similar reasons that improved pastures at Buck Island Ranch have shown higher microbial biomass from long-term land use intensification, despite other studies generally indicating the inverse. And like I said, the wet season was drier than normal, and this may have impacted the trends that we saw. And also, I was expecting to get some CO2 data from the soil that I did not get, but that's something I wanted to look at to see microbe activity. For future directions, if the Rainout project had been going on for longer, we may see different trends. As I said before, we can see if the precipitation, grazing, and the interactions between those treatments would have significant effects. And we could also see, because we had a very dry wet season, if a more typical wet season that's representative of what there usually is would show different trends, such as with that soil fungi relative abundance. We also could perform another sampling event in the dry season to see if there are differences within these communities between the wet and dry season. We could look at the CO2 data from soil, as well as other abiotic factors and soil characteristics to show changes in microbe activity. And we could look at how altered microbial communities scale up to affect plant performance and ecosystem services. So those last two are especially important, I believe, because as stated in one of my introductory slides, changes in microbial communities can alter the rates of nutrient cycling and availability, and understanding how these treatments and their interactions alter or maintain these processes and how plant performance and ecosystem services may be affected as a result could be a great next step in understanding the broader objectives of the Rainout project, which are the extent to which grazing regimes can serve as an adaptation strategy to enhance climate resilience of grasslands utilized for cattle production, and how interactions of different grazing regimes and land management impact the multifunctionality of subtropical grasslands under rainfall variability. So in conclusion, land management, which was the pasture type, had a significant impact on total microbial biomass, relative abundance of gram-positive bacteria, microbe diversity, phosphorus in soils, soil pH, and soil organic matter. Grazing intensity only showed a significant impact on soil organic matter. Most interaction effects had no significant differences on microbe community data or soil characteristic data. There was one that did show a significant result, but it's believed that that was driven by an extremely strong effect of one of the individual treatments for that, which was very significant. And with more time, individual treatments and the interaction of these treatments may show significant differences in microbe communities and soil characteristics. So for my acknowledgements, I'd like to thank Dr. Betsy Boughton helping me with a lot of the coding and the experimental design. I asked her so many questions, even on weekends when I would be working on this. Dr. Greg Saunier, who's not here, he's in France, but he helped me a lot with experimental design, especially in the beginning when Betsy was off. Mandy West, who helped me a lot in the chem lab, and I wouldn't have been able to do that without the knowledge she imparted on me. Carly, who helped me a lot with sending the samples. She's so used to it from the sugarcane project, and always being more support when I was in the lab. Hao Yu for helping me create reagents to use for the nutrient analyses. Luca, a past intern who helped me a lot with those nutrient tests as he was doing a lot, so he had a lot of wisdom to share with me. Claire, who was there for my sampling that whole week I was doing that. She was collecting data for gas for her own intern project. So she was there. We struggled a little bit together for moments, but it was great. Lydia and Emily for setting up the Rainout project and running it. Dr. Zhengxiao Zhu and Haley Henderson at UF for organizing this project. And Akeem, Alma, Chef, and Amartya. You guys are great. Amazing. Thank you. And Archbold as a whole for giving me this experience. So now, any questions? Claire.
What do you think your results would look like, or some of your results, if you took the samples after the hurricane?
Geez, I don't know, maybe the soil fungi would have been different, but also I think that might have caused a good amount of disturbance. So that might have impacted the results we've seen for the grazing intensity. That might have disturbed the soil a good amount. So that could cause, oh, the question was, what do I think the results may have been look like after, if I sampled after the hurricane? So yeah, I think that might have impacted, and we already didn't see trends in the grazing intensity really, but I think that definitely would have just introduced much more variability. That would have been like, okay, there's maybe not something that we can really use. Anyone else? Go ahead.
I was wondering if there might be a correlation, and you mentioned this in the discussion, if there might be a correlation between microbial biomass and an ecosystem service like carbon storage, and whether it might be beneficial maybe to have reduced microbial biomass.
Okay, so the question was if I think there may be a correlation between microbial biomass and certain ecosystem services like carbon storage, and if there may be a benefit to reducing microbial biomass. From what I've seen and from what I know about soil health, usually microbial biomass is always, it's usually a better indicator of soil health the more abundant it is. I think maybe the community structure will depend on that. If it's less beneficial microbes, a lot will probably not be good. But I definitely think it would impact things such as carbon storage, because obviously the more they have, the more they have a higher ability with more to do more carbon storage and certain nutrient cycling and availability.
So there are some questions online. Amanda West says, so does soil fungi in Florida die off rather than going dormant during dry periods?
I can't say for sure if it dies off, but just from what I've seen, there tended to just be not really that much. I know usually they're not going to pop up too much with the very dry conditions. Usually it would be pretty wet in Florida, especially the time I've been here. So we would expect to see them. I don't know exactly if they die off or go dormant, but I know from the soil testing we did, they really didn't find too much soil fungi. So maybe they are dying off.
There's a couple here from Dr. Chu at
Mhm.
UF, and he said, great job. Have you looked into correlations between soil moisture or nutrients versus microbial responses?
The question was soil responses to,
Yeah, like the soil moisture or the
Mhm.
nutrients correlated with with the microbes, kind of like what you did for the soil organic matter. Did you test
Yeah.
any other?
so the question was if I looked at some of the soil characteristics and correlations with the soil microbial variables. I did try to look at some of them. A lot of them were not linear and couldn't be transformed to linear. So it was a little difficult to do that. But from the ones I looked at, there wasn't too much of a correlation from what I saw. And that was, I believe I looked at like nitrate and ammonium specifically.
Anyone else? Lydia has a question.
Mhm.
She said, semi-native pastures have higher plant diversity than improved pastures,
Mhm.
but you found improved pastures have higher microbial diversity. Why do you think the improved pastures, which are more intensively managed and altered, had higher microbial diversity?
So the question was regarding my finding that microbial diversity was higher in the improved pastures compared to the semi-native pastures, while the semi-native pastures have a higher plant diversity, and why I think with the increased land use intensification that may affect that. So there's been previous studies just regarding how certain things like microbial biomass are affected by different variables that I looked at. And a lot of the things I looked at, specifically like microbial biomass in terms of pasture type, were inverse of what general studies were the opposite of what I found at Buck Island Ranch. I'm not sure what exactly the variables are that are affecting that. I think it may have to do with the root systems of Bahia grass specifically that might have something to do with that. I hope that answers your question, Lydia.
And let's see. One more question from John Chow.
Mhm.
For testing grazing effects, was the analysis conducted by comparing responses before and after grazing events?
The question was if for the grazing treatments, it was measured by testing before and after grazing events. We actually have the grazing in the pastures going on now, so I don't believe they weren't measured. I'm not sure when the last one was. I think it was around this time last year. So I only measured I did one sampling event through the week, like the last week of August about. So it wasn't before and after. There was no comparison there. It was just from that previous grazing that had already been done.
And another question from John Chow is, in your linear mixed effects model,
Mhm.
was there any interactions among treatments being included?
Yeah, so the question was if in the linear mixed effects model, I was including the interaction of the treatments. Yeah, I included all of the individual treatments as well as the interaction between different treatments, whether it was like just the precipitation and grazing, just the grazing and or just the grazing and pasture type, and then all of them together. I have some graphs that show, I also looked at that for the non-normal data by doing like the Kruskal-Wallis test, but I didn't see any significant results really in that, except for one of them, as I previously mentioned, but I think that was strongly driven by the pasture type effect. And I think that was in gram-positive bacteria, and I think it was between grazing and pasture type, if I recall correctly.
And one more question from Mandy West.
Mhm.
What is your favorite plant?
My favorite plant. I think my favorite plant is probably a red mangrove.
Great, lots of good job, Kyle. Great job, Kyle. Awesome work. Thanks so much for presenting.
Awesome, thank you.
Okay, thanks everybody joining us on Zoom. Great job to Kyle. Join us next week again for our special distinguished seminar speaker, an introduction to Gunnison Ranch Land Conservation Legacy, presented by Susan Moore and Stacey McPhail on Thursday, October 27th at 3:30 p.m.